It's my great pleasure to introduce Dr. Christopher Gómez today. He is an internationally recognized expert in ataxia and neurogenetics. So he received his undergrad and MD and PhD at the University of Chicago, and also stayed there for residency, and then did postdoctoral research at Caltech with Lee Hood, who we know locally as founder of ISB here. And then he was recruited to faculty at UCLA and then at the University of Minnesota, where he rose to the level of professor and was director of the Ataxia Clinic and laboratory for ion channel disorders and neurogenetics. And then he was recruited to the University of Chicago in 2006 as chair of the neurology department, where he was chair through 2015. So he's still at the University of Chicago. He's an Albina Y-Service professor and the director of the laboratory for ion channel disorders and neurodegeneration, as well as the director of the Ataxia Clinic at the University of Chicago. And so I won't take too much more time and let Dr. Gómez tell us about his work in ataxia.
Thanks, Marie. Let me just share my screen here. So I put this is a new talk. I've never done one like this because I couldn't decide whether I wanted to talk about clinical or research. So I kind of put a more generic talk together. And I want to thank you first for your consideration when I had to cancel on you on April 6th, which is why I put that on my slide that way, which I worked out while in the parking lot of the emergency room where I had just taken my wife, who had an anaphylactic reaction to an antibiotic. That ended up being no big deal, but I was talking to Marie in the parking lot saying, I think I can still do it. I have a room in the ER waiting room where I can give this talk, but ended up being right when she was going home. So we ended up canceling, and thank you for rescheduling. I wanted to talk about what's new and exciting in ataxia because there's more of a spread of new things, and I thought I would talk about some of those changes, new and good. I might even have time to talk about some of our own research if this talk doesn't go too long. So I'm going to give some background for those people who don't do ataxia, a little bit of background in old news to catch people up, and then talk about a couple of new genes that have come up lately that are exciting because they're so prevalent. Talk about a new drug for ataxia and drugs in general. And then if I get to it, I'll talk about some of our clinical research on ataxia. There won't be any of our lab research at all in this talk. That we could save that for a later time. So just as a matter of generic background, spinocerebellar ataxia is a broad family of progressive neurodegenerative disorders, sometimes episodic, that are involved in impaired motor coordination that leads to slurred speech, imbalance, and tremors, and jerky eye movements that are called nystagmus, as you know. And this is due to dysfunction of the cerebellum or its connections. This is a Netter graph, an image of a person probably with Parkinson's, but it looks like ataxia anyways. And here's a picture of the normal-looking cerebellum, a drawing from Netter. I made this image many years ago of breaking down the prevalence of dominantly inherited, recessively inherited, and sporadic ataxias, and recently finally dug down and actually found some true estimates. There are always estimates, but I was actually pretty right. It's about rule of three. It's about a third of them are autosomal recessive, a third are autosomal dominant, and a third are sporadic. And of course, that diagnosis depends on whether or not they found the gene in that sporadic patient or not. It could be changing to one or the other. And I put down hashtags here because those of you who follow this know that there's a number for the SCAs, and there are numbers for two different recessively inherited nosologies, ARCA and SCAR. And there's a small battle going on for people who like to name things between which number and which acronym you get to go in front of the number. They're not always the same. And then this is one of the two slides I borrowed from Hank Paulson, which plots the number of SCAs identified on a yearly basis that he stopped making it in 2008 for obvious reasons because it's exploded. But it highlights very nicely the fact that the initial years we identified primarily expanded repeat mutations that caused ataxia and other disorders, and then eventually we got good enough by either mapping or by sequencing to identify the more conventional mutations like missense mutations and stop codons and splicing mutations. And this was led initially by phenotype studies, like the classical Friedreich's or ataxia-telangiectasia phenotype, or family studies of large pedigrees that leading to linkage analysis, and then validated by either repeat expansion hunting or DNA sequencing. And eventually tools were developed, as you know, for direct DNA sequencing, depending on whether you can match the phenotype or just get lucky and identify a novel mutation or repeat expansion. And of course, to me, what's fascinating is follow-up studies in many, many cases led to a broadening of our understanding of the phenotypes of these disorders. And I'll give a couple of examples of those. But here's another way of depicting ataxias. I thought I would borrow this from the McDonald's Corporation because we've had well over 50 SCA types, 50 SCA genes, and we actually screened for 650 different ataxia genes in our exome sequencing. So it's way over the top and impossible to graph anything but golden arches for this. So I also borrowed this slide from Hank Paulson, which is a great way of depicting the importance of expanded repeats in neurological disease. This is kind of a model gene with a coding exon, a 5' and 3' UTR, and a couple of introns. And most of you who know genetics know this story very, very well. There are a number of CAG repeat expansion disorders, the polyglutamine ataxias, the Huntington's, SBMA, and DERPLA. There are also 5' UTR repeats, both CAG and other intronic, other 5' UTR repeats such as DM2. There are CTG repeats in the 3' UTR such as CA8 and DM1, and there are pentameric and hexameric repeats in introns that cause ALS, familial ALS, SCA31 and SCA10, and then Friedreich's ataxia is the first intronic repeat identified, causing which is needs to be a biallelic repeat expansion. And soon after that, cannabis was discovered, which I'm going to talk about, a biallelic pentameric repeat. And then recently, SCA27B, a dominantly inherited GA repeat expansion in an intron of a gene that is already known. And FAXTAS, which is in the 5' UTR. And I actually left out one of the hereditary epilepsies that has a large repeat tract in the 5' UTR. So I think as neurologists, we should be proud of the fact that this is our domain. There are pretty few other disorders that are caused by repeat expansions. There are some, but not as many as this. And it's really up to us and our collaborating molecular geneticists to try to figure out a way to silence or reactivate the consequences of these repeat expansions. This is a graphic slide, a table slide that I think is ridiculous for two reasons. One, it is too hard to read, and the other is that it's way out of date intentionally. So I showed it this way just to highlight these complex names we have for these disorders, the genes, the proteins affected, and the range of clinical symptoms, but also so I could add the very first genetically defined ataxia, Friedreich's, and cannabis that I'm going to talk about, which is the most recent genetically defined recessive ataxia. And below I put why this is ridiculous, because in 2018, Sanovic and Nemeth reported in the Handbook of Clinical Neurology 32 types of recessive ataxia. And soon after that, Nicholas Dupre reported in a classification 58 different types of recessive ataxia. And as I said, we look at 650 different ataxia types in our exome sequencing panel. So here's Friedreich's ataxia. This is a classic disorder, clinically definable in the childhood form as presenting with areflexia, sensory ataxia, cardiac involvement, and skeletal changes in diabetes. The mutation is, as I said, is a GA repeat expansion in the first intron of a 5 exon gene called frataxin. The expansion goes from 100 to 1800, and there's a big variation in the severity. the repeat causes genetic silencing, transcriptional silencing of the disease, of the gene. So there's reduced mRNA and protein. And about 4% of these patients have a biallelic mutation with a 1 repeat expansion and a point mutation in the frataxin gene, causing a slightly different phenotype of the disease, with often spared reflexes. So that's one example of expanding phenotype. And here's another example of expanding phenotype, and I'm not going to play this video, but this woman is one of many patients I have who have age of onset after the definable age of Friedreich's. Her onset was at age 31, and I've had patients who have onset in their 50s, and Subramoni reported 2 patients who had onset in their 60s. These patients typically have no cardiomyopathy and no scoliosis. Her exam showed dysarthria and tension tremor in coordination and hyperreflexia. So it defines what Anita Harding called late onset Friedreich's ataxia, or FARR, Friedreich's ataxia with retained reflexes, which often she would find in other kindreds, where some siblings would have the classical presentation and cousins would have a milder presentation because the repeat expansion was different. And so this patient showed what we see in this disease is silencing of one of the alleles with 843 repeats, and relatively modestly affected transcription in the gene, and the other allele with only 101 repeats. Subramoni's patient had the patients had 101 repeats in both alleles, and they had interruptions. So that led to a very, very mild phenotype, and these patients were still walking in their 70s.
And how old?
So in comparison to that is the most recently defined recessive ataxia, which is called Canvas. And I'm going to tell you that I heard of this disease from a number of neuro-otologists who contacted me and told me about this new disease that they had seen. And I really didn't believe it at all. I was skeptical because the patients had cerebral ataxia, which all my patients do, neuropathy, which many of my patients do, and vestibular areflexia, which I suspected many of my patients had. I think my SCA3 probably do, but I didn't have proper tools to test for that. When it was published and recognized that they had this chronic cough, I realized that I was sitting on a lot of patients who had this disorder, Canvas, which is a new disorder, which is characterized by biallelic expansions of an intronic repeat in this gene called RFC1, which is a replication complex for DNA polymerases. And I neglected to mention in my recessive ataxia panel, the other reason why I use that table of patients, because about 6 types of recessive ataxias are due to DNA damage and repair disorders. And this gene is also involved in DNA repair. So this added a 7th one. And Cortese reported 29 individuals with this disorder, and it really launched a whole new field of research. And it also is a great example of how, to an increasing extent, for reasons that I'm not fully clear on, the Canadians, Europeans, and Australians are really getting into the act much more nicely in terms of gene discovery and research in ataxia. This came from Henry Holden's group in Queen Square. So of course, the spectrum immediately began to broaden, and you'd see patients who have more of a predominant cerebellar phenotype and subtle inklings of the other features, sensory neuropathy, and these pieces are picked up in neuromuscular clinics, and vestibulopathy, and these patients are being picked up in neurotology clinics. And my ENT colleague, who just recently published a paper with me on characterizing the cough, picks up these patients when they come to him with chronic cough. And as you might expect, just like in Friedreich's, as we began to look at this classic phenotype, we would do an exome sequencing in patients who had a monolithic expansion, and a small number of them, about 1%, have loss of function mutations in the RFC1 gene with the classic picture and chronic cough. The cough also slipped by me because I thought everybody with ataxia chokes and has cough because of that, but it's really quite distinctive. And they have, when we investigate this, they have an amyotrophy of the cardiac sphincter due to probably vagal nerve involvement, and they have chronic reflux leading to a suppression of the cough reflex, of the sensitivity of the leading to the cough reflex, and it can precede the disease by years, precede the onset of the ataxia by years. Now switching to autosomal dominant ataxias, this is another ridiculous table of ataxias that I made many years ago, but I use it still to highlight some of the overlaps and similarities. These are the ones that are caused by CAG repeat expansions, such as SCA1, 2, and 3 in the coding region. This set are characterized by non-coding repeat expansions, and I haven't even updated the SCA27 one on here yet. This grouping are some of the conventional mutations in other genes that are usually critical genes for neuronal functioning, such as the spectrin gene or many ion channels. And interestingly, SCA27, a little known ataxia caused by coding region mutations in the FGF14 gene, is the same gene as affected in SCA27B, but a different kind of mutation, another non-coding mutation. So to talk about a little bit about the anatomy, many of these ataxias are multi-system disorders, really much like an MSA, affecting many motor nuclei. But SCA6 was and has been really the canonical pure cerebellar ataxia, affecting only the Purkinje neurons, as depicted in this graph, and it's caused, as you know, by repeated expansion in the calcium channel gene CACNA1A. And it's characterized by atrophy of the anterior vermis, leading to of the cerebellum, leading to due to loss of Purkinje cells. It's characterized by downbeat nystagmus, cerebellar atrophy, and Purkinje cell degeneration. And here's a picture of some empty basket fibers in a Bielszowski silver stain, which is what you typically see. So along comes a new ataxia, just defined literally 7 months ago by the Quebec group led by Bernard Brace, as a new form of ataxia due to intronic repeats in the same gene as SCA27. And they originally they called it SCA51. But then I don't know who was in charge of this, probably the Hugo organization, decided to name this SCA27B because it's the same gene and probably the same mechanism. So here's how they did this. This is really an exciting paper and exciting work. They had 3 kindreds in the Quebec collection that had classic late onset cerebral ataxia, episodic features, and downbeat nystagmus, which looked much like SCA6. And they tested negative for SCA6 and all other SCAs. So they invested in whole genome sequencing for 6 of these patients. And lo and behold, they found that they all were heterozygous for a large GAA repeat expansion in the FGF14 intron in one of the predicted transcripts, transcript 2 of FGF14. And they went ahead and developed sequencing PCR primers for long-range PCR and confirmed that these patients had expansions of one allele of the FGF14 variant 2 in several of the patients, and normal size of the other allele. And it had defined what's turned out to be an intermediate zone set of repeats. Patient 3 here was symptomatic with 283 repeats in his expanded gene, but control patient FCC57 has a repeat of over 280 and is so far asymptomatic. So they define an intermediate zone of over 250 and under 300, which is recognized as the intermediate range that is a variable penetrance. And here's the graphic of that done with a density of repeat numbers. The normal population of 800 chromosomes shows some of them between 250 and 300, which at the present time were asymptomatic in the normal population, and 128 chromosomes from the affected population. Of those, there were 122 that had normal repeats and 134 expanded repeats, which means that several of them had 2 repeat expansions in the either intermediate range or expanded range. And that was done when they began to collaborate with the German group, the Australian group, and a group from India. which they correlated all of their DNA samples and recognized a range of prevalence from 10% in the Indian group to 61% of the affected patients in the Canadian group. These are, of course, are highly curated populations of patients, so it's unclear what the true native prevalence is. And then this panel from that paper is a little bit out of place, but it's the most interesting panel I've seen of this whole paper because it shows the interparental stability in meiotic events from either male parents or female parents. And what's very interesting, and it's been mostly true in our hands, is that the male parent transmits oftentimes a reduction in size of from a few repeats up to as much as 200 repeats, assuming the paternity has been confirmed on these patients. And the female parent transmits an expansion of up to 100 in some cases. And we have seen this in our grouping. Our friends in Michigan have actually found there's some violation of that trend in some cases. In our Chicago group, we first tested these of our 126 patients from my collection from Chicago and Minnesota. 23% had expansions in the pathological range. So this is the clinical panel, which I wanted to highlight. That's just a couple of interesting features that are relevant if you think about these ataxias like I do. Their age of onset is just like SCA6 and other late onset ataxias, between 59 and 62 years of age. There's episodic features in a high number of them, from as many as 70% in some cases, episodic features. Sorry, as many as several percent in these patients. In our case, it's about 30%. The median age of onset, however, is very interesting. It's late onset. It's something actually I explained to Alexander Doerr many years ago, that I thought many patients presented with a late onset of episodes compared to the progression of the disease. And that was distinct from the classic disease EA2, in which the onset of episodes is much younger than the progression of the disease in that CACNA1A disorder. The triggers, however, are the same as EA2. The exercise, alcohol, caffeine, sudden movements trigger episodes in this disease, just like in EA2. They have nystagmus, much like SCA6, downbeat nystagmus and gaze-about nystagmus, like many of our patients. What's very interesting is there's a large number of patients who seem to be sporadic, and this could be due to the changing of the repeat size between the different gender transmissions. This is a figure that shows this correlation of inverse correlation between repeat expansions and age of onset, which is roughly present, although there's a large spread using repeat expansion sizing and estimates of age of onset. The pathology is just like SCA6, cerebellar atrophy, anterior vermis atrophy, abscess of Purkinje cells. Here is one torpedo. I think those are reported in many different ataxias. This is a comparison to the normal Purkinje cell layer in panel E. And this is to show the empty basket fibers and total absence of Purkinje cells in a silver stain. And this is some of our results that we were able to do after we canceled on our April grand rounds. This is the patients that we were able to collect from three groups: Chicago, UCLA, and University of Minnesota. 91 patients so far in our cohort, and the repeats are both in the intermediate range, and 31 were in the intermediate range, and 60 were in the expanded range. And it's the same findings as the original paper with family history in many cases, but not all. Prevalence of cognitive impairment in some of them. A statement about progression of disease from 10 years to using a walking stick, and 15 years to using a wheelchair, which is actually quite similar to other ataxias. So it's a later onset, but progresses at around the same rate. Prevalence of the same number of neurological findings, with unsteadiness in episodes being the highest prevalent thing for an age of onset, all around the same at the same time as reported by Bernard Braze's group, and a high prevalence of episodic ataxia in our patients. Interestingly, they go back to baseline but not in all cases. I'll just skip over the ocular findings and the exam findings and the MRI findings. So for this very interesting disease, what is the effect of the mutation? As I said, it's a repeat expansion in a non-coding region of the FGF14 gene in one transcript only, but it causes reduced protein and mRNA. So, reduced protein, but not reduced mRNA. This is the expression study from the cerebellum of these patients, and both transcript 1 and transcript 2 are normal in abundance, even though the repeat expansion is shown here in the part of intron 1 of transcript 2. But protein levels are reduced in Western blots from cerebellum of these patients. And when the investigators were clever enough to make iPSC-derived motor neurons from these patients, the FGF14 protein is reduced in these patients, and these are not antibodies that can distinguish between the two variants of this protein, two different forms of this protein. So it shows reduced protein levels due to this repeat expansion. So what is this protein? There are two of them. There's an intracellular form and a secreted form. And the most well-known form prior to this well-studied form was really the secreted form, which is a 247 amino acid protein that's involved in mitogenic activities, survival activities, and a variety of cellular processes involving cell growth. Not relevant, I think, to this disease. But there's also an intracellular form, which has been called variant 1B. And this form is involved in organizing a number of ion channel membrane insertions and stability, including NAV 1.6, KCNQ, and CAV 2.1. And I should say that a lot of this work was actually Vikram Shakatai's postdoc work with Daniel Ornitz when he was in WashU. So it's kind of actually PhD work, which was kind of interesting. And he did this work, I believe, around the time the SCA27 was discovered as a effect mutation of the FGF14 protein. So FGF14 knockout mice have these electrophysiological findings of decreased excitatory transmission in the parallel fiber synapse and reduced AMP receptor excitatory EPSCs and reduced glutamate transporter functioning. So that would mean they presumably have abnormal VGLUT1 staining of their fibers, and so a number of transmission changes that lead to an effect of the actual complete knockout of these mice. But it may well be that they were studying the effects of the intracellular form rather than the secreted form of this protein, which was not known at that time. So in summary, SCA27B is a new late onset hereditary ataxia, very abundant. It's a pure cerebellar disorder. It's often has an episodic pattern, but different from the episodes that occur much younger in children with EA2. And there's an unusual male parental transmission that can lead to skip generations. It really mimics SCA6 in many ways, and I think this could merge with the ion channel aspects of SCA6 and CACNA1A disorders in general. But because of this loss of function feature, FGF14 restoration of function may be a very rational therapeutic strategy for SCA27B. And I think that there are probably many people in laboratories thinking about how to confirm this in preclinical models. So lastly, I want to turn, or not lastly, but if maybe I may have more time to finish with something else, I want to talk about some drug studies. And this most important trial that was just published in 2021 by David Lynch's group, highlighting the safety and efficacy of omaveloxolone in Friedreich's ataxia, which is called the MOXIE study. And that led to, most amazingly, in the last few months, approval by the FDA of this as the first treatment for Friedreich's ataxia. And they used 103 subjects for this study. And the background for this, I'm going to review a little bit here because the preclinical studies really led to some insightful efforts to get the right track. The first experiment that was done in Friedreich's ataxia when the gene was discovered was a knockout of the delta F or the YFH1 homolog of frataxin in yeast, which led to a poorly surviving yeast with iron depletion of the cytoplasm and iron overload of the mitochondria. And that led to a flurry of work on free radical damage, et cetera, a recognition of iron deposition in the myocardium, and studies by Martin Delaticki of iron overload in the fibroblasts of patients with Friedreich's. So to summarize, a lot of that work with these 5 bullets, frataxin deficiency is a result of a repeat expansion, not unlike the canvas repeat expansion affecting the RFC1 protein. But if you silence this protein, you see accumulation of mitochondrial iron, oxidative damage to a large number of macromolecules, sensitivity to additional oxidative stress, impaired mitochondrial energetics, and most interestingly, impaired behavior of this protein called nuclear factor erythroid 2 related factor, or NERF2, which is a signaling protein which enters the nucleus during stress and mediates ATP production in response to oxidative stress. So in some of this work, healthy fibroblasts, this protein translocates the nucleus to activate antioxidant genes. But when FA fibroblasts are studied, these actions are dysregulated and it does not translocate properly to the nucleus. And so this protein was discovered, this molecule was discovered as a potent NERF2 activator called OMAV, or omavaloxolone, which actually restores function in Friedreich's cells and FA mouse models. So this was important preclinical work to lead to the launching of must have been the 10th clinical trial on Friedreich's, given that they've used frataxin activators and free radical scavengers and a variety of other molecules that failed. This led to the launching of a clinical study, which is the OMAV treatment trial. And this is the protocol for the study. They define as the primary outcome a clinical rating scale called mFARS. The mFARS provides a quantifiable measure, semi-quantifiable measure of neurological function in Friedreich's patients, and similar to what you all may know as the SARA, or the Scale for Assessment and Rating of Ataxia. It's only slightly more quantitative, but it has very similar features. It has 4 subsections: bulbar, upper limb, lower limb, and upright stability. And they set up a 52-week protocol with randomized placebo trial, and the examiners were blinded to the treatment and also the lab results. And they had some other secondary outcome measures which did not track so well with the results. But as far as ADL, the FA ADL measures were somewhat validated, and a number of other things like 9-hole peg tests and time 25 walk, frequency of falls were used. And in this study, they used the Friedreich's, the mFARS trial, and this is just a screenshot of one of the panels of why this semi-quantitative study tool may be better than the SARA in the fact that it uses a lot of quantifiable upright stability measures. That is, they time the several ways of testing stance, with feet apart, with feet together, with feet with eyes closed, and with feet in tandem stance. And they give 3 trials to do this. We use the same trial ourselves in our study, and it's burdensome, but a little bit more quantifiable than the SARA rating scale. And this is just the final summary slide of this study, which showed that when patients were enrolled, the placebo patients continued to worsen up to a matter of 2 points over time, whereas there was a protection and a stabilization of the patients who got OMAV over the 48-week trial. And this was the same thing that was seen when they parsed out and stratified for just the upright stability measure, which was the most informative portion of the Friedreich's of the mFARS study, that confirmed the efficacy, or suggested the efficacy of this drug. And that alone was what launched this into FDA approval. So I don't know how many of you who study ataxia are now using this drug, but I have now enrolled about 8 patients with the commencement of this drug in the last month or two for my Friedreich's trial. So in summary, the OMAV drug, which is now called Skyclarys, and I don't know why they chose that name, was approved by FDA and it seems to slow progression. And on the other hand, most recently, in the last 2 or 3 months, FDA failed to approve the drug troriluzole, which is a riluzole precursor. That there was a lot of money was invested in trying to study this drug, and we were involved in this trial, and this trial failed. And most recently, and I know Marie was involved in this trial, the ASO trial for STA3 was stopped in the preclinical phase because of preclinical toxicity molecule. So there's a lot of hush-hush about this, but this trial was stopped in the preclinical phases. So that's the update I have on drug studies for ataxia.
Sorry, Chris, you cut out just right when you were saying about the FDA for the ASO trial.
At which stage?
Just what the FDA had stopped the ASO trial and then
Yes.
sorry.
Okay. So to what, I mean, FDA was not involved in that. There was the company Biogen, which was running, as you know, Marie, the ASO trial. They stopped this in the preclinical phase of the disease because the Ataxin 3 ASOs were up. This is by rumor only. The Ataxin 3 ASOs were toxic to the monkeys in the preclinical phase. So they abandoned the trial, and the data for that have not yet been released as far as I know. So that trial has been stopped. And really ASOs in general are kind of on right now because of a couple of trials. Despite the success of the SMA trials, a couple of trials for the repeat expansion diseases have now been stopped. Did you hear me okay on that time?
Yes.
Okay. All right. So let me get on if we have time for a few more slides on our research, on some of our clinical research in the ataxias, specifically on our ability to try to attempt to measure digitally performance and coordination and frontal lobe function in the ataxias. You might have gathered from the Sarah scale and the Friedreich scale that these outcome measures are very, very crude and only semi-quantitative. So a number of years ago, we got into the use of digital wearables as a way of measuring different aspects of performance and function. And I collaborated with the group at OHSU to do this. And in 2017, I launched this project with them and in Chicago, trying to measure ataxia using wearable sensors. And I'm going to talk, we published a number of papers on this, so I'm going to talk mostly about the even more lofty goal of attempting to measure ataxia in the preclinical state, in the pre-manifest state, because it's our belief that the preclinical state is a period of maximum preserved nervous system function that would allow us to potentially treat irreversible treatment before irreversible changes set in. So it's the best time to devise therapeutic interventions, and it may also reveal pathophysiology and ataxia compensation. And because of gene testing, we have the ability to launch these questions in the preclinical state very easily now. So I wasn't the first one to be involved in this by a long shot. The European groups tried to study the SARA in the preclinical state, and many other groups used gait, ride, and eye movement studies, cortical spinal tract activities, and MRI spectroscopy in patients who had a positive gene test but had no clinical signs of ataxia using the SARA score. So we applied these wearable sensors for this purpose, and this shows an image of somebody wearing many more sensors than we typically use on their bodies. And these sensors are accelerometers, they're gyros, and they're interferometers that look at RF signal for rotation and communicate with each other. And so they allow assessment of a large suite of gait and stance movements. This shows a patient walking using some of these sensors on their chest, their waist, their feet, and their wrists. So it allows us to measure both mean changes in a value as well as the variability of that value. And I'm talking about many different foot and gait and stance values. So we weren't the first ones to apply this to ataxia. Our friend Winfried Ilg in the German group studied this very early on in 2016, a year before I started, and looked at tandem gait and using our sensors, using the sensors from the Portland group, and found that even in pre-manifest patients using challenging tasks like tandem gait, the coefficient of variation of step length was predictably correlating with the number of years before age of onset of the disease in a group of SCA patients. So this is the first use of these wearable sensors. And then when we started doing this, we arranged to collaborate with the Cuban group, who were really rough and ready to go. And I translated the tasks for these tools into Spanish and sent them to our colleagues, and actually flew there and taught them how to use them. And they studied 30 SCA2 pre-manifest patients, and they used tasks such as gait and stance, and were able to apply the prediction of age of onset and a SARA score of under 3 for their patients. And they found, again, variability as the prominent finding. The variability of the toe out angle and the variability of the swing metric was statistically significantly abnormal in patients who did not have any clinical signs of ataxia. So variability of gait timing is a key feature in the ataxias, gait performance and stance. So then this is our work in Chicago and Portland. This is 163 SCA patients who have clinical SCA, and we use the area under the curve to separate out a number of metrics that would predict differences between control and patient. And the red bars are those values that were variable values, and the blue bars are those that were actually mean values that were different. And you can see the prominent feature is that many of these values that we measure show higher variability than the control. Up to as many as 10 different values are different from control in these patients. This is a precursor to establishing this as a metric for use for progression of the disease. So then we took a number of these patients who were pre-manifest, and we found, although the prediction was not as strong, only values of 0.6 for these values, they were the same values, such that all of these things were trending towards becoming the predictably more variable values as the patients went from symptomatic from pre-symptomatic to symptomatic in a cross-sectional study. So the prominent feature in these patients is that individuals with pre-manifest ataxia, in many measures, there's with normal SARA scores, have increases in variability of many gait and stance parameters that overtly become abnormal in patients with symptomatic SCA. So how can we use this for something even better than that? Measuring variability will be useful, and we'll be able to plot that over time, but we wondered why they're variable and what was going on in the rest of the brain that would reflect that. And so we studied something, we studied the phenomenon of walking automaticity. And all of you know about automaticity in an unconscious fashion. When you walk downstairs and talk to your friend walking downstairs, or when many of us walk across the street in front of cars while looking at our iPhones, we are taking advantage of the fact that walking is an automatic process which breaks down when the central pattern generators of all sorts are degenerating, such as in Parkinson's or in ataxia. And we wondered whether or not there was a reflection of the frontal lobe control that would be manifested in the case of a breakdown of walking automaticity. This is actually known by Sherrington, and this is the Sherrington cat, which this cat has no contribution of the upper motor pathways. This is only vestibulospinal tracts, allowing this cat to walk on this treadmill of increasing speed because he's had an intercollicular transection, and they walk normally. So the clinical significance is this breaks down in all kinds of motor disorders. And so we tested the hypothesis that prefrontal activity would have an inverse relationship to gait automaticity in the ataxic state, and may be a means to detect the earliest signs of loss of gait function in the pre-manifest state, such as occurs with increased variability. And this could be an ideal indicator of a therapeutic window. So we did this using basically an ambulatory form of functional MRI, which is called functional near-infrared spectroscopy, in which the patient wears a device called an Octomon, which compares levels of cortical oxygenated hemoglobin levels in different parts of the brain. This is only used in this case in the prefrontal cortex. This is all done by my collaborator Martina Mancini, who really has applied this to a number of different types of patients, and with me with ataxia. So this device uses one receiver on each side of the hemisphere, and two types of transmitters. A transmitter with short separation, which is used to average out noise, and transmitters of long duration, the ones in the dark red dots, that pick up changes in oxygenated hemoglobin after subtracting the short separation. And the tasks are a normal 2-minute walking task, and then some walking around a chair in circles, and then walking using some distracting auditory signals. And what Martina has found, was that there is a change in level of prefrontal activity in patients with both manifest ataxia and pre-manifest ataxia that is significantly different while walking. And this is a raw data graph of 3 different individuals, one control in blue, and a pre-manifest and a manifest. The first thing you should notice is that in the control, what happens after initiating the walk, after 20 seconds of standing, initiating the walk, the patient rapidly returns to a low level of prefrontal cortex activity, because the patient is doing an automatic function of walking, normal walking, without any neurological impact. Whereas the patients with ataxia develop an increase in prefrontal activity that continues abnormal in the symptomatic state throughout the entire course of the activity. But even more interesting is that the patients who have pre-manifest ataxia, the level of prefrontal activity involvement continues to increase through the entire task. And also interestingly, this patient is turning back and forth over a 10-meter walkway, and each time they turn, we believe this peak is a sign of their increased prefrontal activity during turns. And we can plot that because they're wearing wearable sensors at the same time that will measure their turning activity. And if you break this down into the early stage in the box here and the late stage, you can see that the big changes here are in the pre-manifest state. Is the late stage continues to have increasing prefrontal activity in the pre-manifest, whereas the manifest, there's a great increase in the pre-manifest in the early stage of the task. So we think this is a great reflection of a stage of the disease that is overactivated, in which the prefrontal cortex is overactivated in an attempt, we believe, to compensate for the disease. And we think that as we begin to test patients who are earlier and earlier with their positive repeat expansion, but no disease, we will identify a normal stage in which there's no increase in the prefrontal cortex activity. And that window, prior to the onset of PFC activity, is the total pre-symptomatic period. And those that have the increase in PFC activity prior to the onset of symptoms is a true pre-manifest state of a prodrome prior to the onset of symptoms of ataxia. And we tend to target these patients for disease modulating activity, modulating drugs. So in summary, prefrontal cortex plays a dynamic role in gait fluctuations in people with SCA. The increased prefrontal cortex activity while walking in both symptomatic and pre-manifest ataxia patients may indicate compensatory responses that we can use to target therapy. And a return to gait automaticity is an early failure to return to gait automaticity, may be an early hallmark of pre-manifest ataxia. And we hope to use ambulatory FNIRS as a powerful tool to characterize this pre-manifest state in ataxia. And here are the collaborators for all these recent studies I mentioned. This is Fay Horak's group, with whom I have been collaborating now since 2017 in the OHSU. This is James McNames, who is a engineer and mathematician in the APDM group and a faculty member at Portland State, who is a co-founder of the company APDM with Fay Horak. This is Martina Mancini. She is the scientist who worked on the PFC FNIRS activity. My coordinator in Chicago is Hannah Casey, who arranged all the entire idea study with us. Widad Abuchar is my movement disorders fellow, who did the clinical work on the SCA27B, and Mohammed El Gohari is the engineer who helped me launch the sensor study for ataxia patients at APDM. And I'll take any questions you have there.
Thank you so much. That was a really great talk with a great overview for ataxias and a lot of cutting edge updates. So that was great. I know there's some questions in the chat already.
Oh,
So,
I see Tom Bird's hand up.
yeah.
Is that a chat, or is that?
Hey,
Yeah,
Chris,
hi, Tom.
this is a fantastic talk. I enjoyed it a lot. I had a quick question for you. I couldn't tell if it's known why the protein is decreased in the FGF patients.
Well, if I know these guys by now, they already know, but I don't think that that this has been talked about yet. I probably could email David Pellerin. We've been busy doing other things, but they're moving so fast and so well that repeat expansion might might be involved in some translation abnormality of some sort. I'm not sure. It certainly doesn't seem to, or sorry, splicing abnormality, but it certainly doesn't seem to affect, able to affect translation, since it's removed from the transcript. I really don't think we know yet. I'm sure that they are hot on the trail of figuring that out.
Yeah, good. Thanks.
Mm-hmm.
Chris Ransom has a question in the chat about I think it was going back to the FGF14 patients, how long their episodes are. Are they on the order of hours, days, or months?
Yeah, I did skip over them. It was actually Widad's study. I don't think that the Canadian group had that information. So we wanted to capture a few fun clinical things as we were sort of following up on this. We were being the latecomers to the field. So Widad has that result. So is this it? Yeah. Duration, yes. Between 1 and 15 minutes, 16 and 30 minutes, 31 and 60 minutes. So they last much like EA2, many of them over 90 minutes. Okay, so to me, it covers actually the spread of EA1 episodes and EA2 episodes. Hi, Chris. Chris, I know your dad. So nice to meet you.
And then Dr. Weiss, Mike Weiss has a question about looking at pre-manifest patients to see if you can track neurofilament or CSF or other biomarkers with like when they like a certain threshold or percentage increase to develop symptoms. And he says,
yeah, I don't know that person is on some of the Ataxia Global talks, but there was just a talk about 2 months ago, 1 month or two ago. They're looking at that in the German group. They're looking at CSF neurofilament levels, and there is a rough correlation. I think that's what you're asking about. That I'm sure this has not yet been done with the pure cerebral ataxias. The new one, I should say, 27B. We did look at it, look at some of these levels in SCA6 CSF, but we didn't have anything that we thought was publishable. I'm not sure that's going to be a very useful outcome measure and biomarker, but people are working on that in the European collaboration.
Well, and he also asked, are there any SCA studies ongoing? I would say CRC SCA, hoping to find biomarker
Are there any SCA studies ongoing?
for looking at for trying to identify biomarkers in SCAs.
Yes, I mean, the one I just talked to you about, if you could call our sensor studies biomarkers or performance outcome measures. Yes, the lead person in that is Puneet Opal in the CRC SCA group. There's a triumvirate of leaders in that group: Liana Rosenthal, Vikram Shakata. Actually, it's now 4. Puneet Opal and Shenghan Kuo are the 4 leaders of that group. We're actually meeting next week in Minneapolis for a investigators group. If you're interested in being involved in some things like that, you should email me and I can connect you with the people in the National Ataxia Foundation, who are a very well organized group.
So we're a site. We're a site.
Oh, congratulations on that. You
And I'll see
received
you
your
next week.
paycheck of $25,000 yet? I'll see you next week then. That's great. I'll
Yeah.
meet you face to face, Marie. That's good.
So it looks like Sumi has a question.
Hi, thanks. Fantastic talk. Thank you. The prefrontal cortex study was really interesting, and I can imagine it, you can invoke the planning piece of prefrontal cortex. That all makes sense. But do you think that it gives us any insight to maybe the altered the altered cognition in some of the ataxias, or give us insight into psychiatric features? Does it tell us?
I mean, I would say, I don't know. I'm not a big believer in this. This is a Jeremy Schmaman belief. But I would say I have circumstantial evidence that this plays no role in that most of these patients were pre-manifest. Okay, and so they have normal functioning entirely, and no signs of any cognitive problems at all. They're living, working people. So the abnormalities that we see as heightened in the PFC FNIRS activity don't seem to reflect any signs of cognitive problems. Now, I didn't mention what Martina did as part of the study because it's part of her Parkinson's study. She used this really interesting auditory algorithm where the patients are forced to be distracted by listening for letters and listening for particular letters, and then count the number of times they hear an A, for example, wearing headphones. And I think she gets a lot of mileage with that, with the Parkinson's study that she does, but not, unfortunately, for the SCA patients, which maybe also adds into that.
Right. Interesting. Okay, thanks.
Well, I had a question that I think is more for you to kind of expound on hypothetically. It's always been I've always wondered why, is there something about the brain that repeat expansions, the brain's more vulnerable to, because a lot of these genes are expressed all over the body, and so why is it more?
I might not want to do that with Tom Bird in the meeting, because he might shut me down for being so. I do not know. I do not know. It could be, the thin connection between the DNA repair disorders comes into play, because 6 or 7, I wanted to mention that, it's AT and AOA1, AOA2, ATLD, and then now RFC1, sorry, and now FGF14, all involved in DNA repair. And then, of course, there's this amazing story of somatic expansions and patients with HD who have variants in their DNA repair repertoire of genes, who have further expansions in somatic expansions that may play a role has yet been unidentified in the ataxias. Benendiani did some in Friedreich's, but why are all these cells vulnerable? All these neurons vulnerable, including motor neurons in C9orf disease. Could it be that our somatic cells are much better at repairing DNA because they're not in terminal differentiated state? Could or some reason like that? I don't know. I do have one postdoc who may be chasing that because because we're doing single cell and RNA-seq on Purkinje cells and granule cells. We don't have the perfect model, but that may be a way we can look at that. And you might look at Al Espada's recent paper where he did it with SCA7 mice, and they have and it's a targeted integration of the CAG repeat expansion in Ataxin 7. So he may have come up with some transcripts that are sort of are spared in granule cells and unspared in Purkinje cells, for example.
Ooh,
So Marie, I think there are two pieces to it. One is when the repeats get too long, that's bad news. That's bad for the brain.
Mhm.
But the repeats, when they're not too long, I believe have some kind of evolutionary brain development advantage to humans. Lower animals don't have them at all. For mice don't have repeat expansions
Right.
normally, and humans do. And so I think it's evolutionary, and I think as long as they're not excessively expanded, they're actually somehow being productive to human brain function. However that is, I don't know, but that's my suspicion.
Mm-hmm.
So Tom, you let me babble on. You had a better answer than me.
No, that's really interesting. That's
Any answer to that is a good answer, because no one knows.
Well,
Well,
the late Sue Lindquist had some thoughts about repeat expansions and how they would somehow provide some of that protein conformation memory in gene expression. Unfortunately, I never got to get her to expand on that any further, but Aaron Gittler, who was a descendant of her, might be able to explain what she meant by that.
thank you. Thanks so much for that great talk and great discussion. I think we're up to the hour, so I
time.
don't know if we have
Out of
any
time.
questions, but I'm sure I can ask you more questions next week too.
I didn't know you were going. I'm really looking forward to seeing you there, and anybody else who goes. Yeah, it's got to spread the word. We are a very small field, and we need more young people like you, Marie.
Great. Well, thanks. Thank you so much.
Email me any more questions. Thank you so much. Take care, everybody. Thanks for the invite.
Thanks for the wonderful talk. See ya.
Okay. All right. Bye-bye.